A research review presentation on the 2008 study, Speech Production in 12-month-old Children with and Without Hearing Loss. 3 researchers conducted a study that was published in the Journal of Speech, Language, and Hearing Research in 2008 titled Speech Production in 12-month-old Children with and Without Hearing Loss. The purpose of this study was to examine whether early identification of hearing loss, early amplification, and early intervention facilitate the speech development of children with hearing loss at 12 months of age. In other words, the purpose was to compare speech production of 12-month-olds with and without hearing loss in terms of syllable shape, consonant type, and vowel formant frequency. A literature review was conducted and composed to support a view of early speech development in which infants begin modifying their vocal tract gestures during their first year of life to resemble those of adult speakers in their language community. The literature review also describes ways in which speech development and production of young children with hearing loss differs from children with normal hearing. Research supports the fact that linguistic environment influences young children. Researchers use discrete formant frequencies from vocalic segments as evidence that infants produce babbled vowel-like sounds in language-specific ways. During a previous experiment, the same researchers determined that infants started to incorporate language-appropriate postural adjustments into their babbling based on discovered similarities between long-term spectra of infants' babbling and speech samples from adults in their respective language communities. Research also supports the fact that F2 analysis is meaningful in determining linguistic influence. Researchers have provided evidence to support the hypothesis that vowel formant frequencies, particularly F2, can be used to explore the extent to which the language environment has affected very young children presumed to be at risk for delays, such as children with hearing loss. Research supports the fact that early hearing is important for vowel acquisition. A 1996 study concluded that children with early onset otitis media, and therefore a conductive hearing loss, were delayed in acquiring the extent of tongue placement in the front-back dimension as measured by F2 standard deviation, which is evidence to support the importance of early hearing to vowel acquisition. Research supports the fact that vowel formant frequency analysis is meaningful in determining transition from babbling to speech. Research suggests that jaw oscillations provide the frame for the tongue and lip content as an infant progresses from babbling to speech. Greater tongue control to produce vowels should result in greater front-back movement of the tongue, and therefore greater F2 variation. Therefore, vowel production, in terms of tongue position and formant frequencies, can be an important indicator of the transition from babbling to meaningful speech. And lastly, research supports the fact that utterance shape is different between children with normal hearing and children with hearing loss. Researchers have observed that children with hearing loss are delayed in producing canonical babble, which by definition must have consonant-like margins accompanying the vocalic nuclei. The participants of the study included 20 children, all 12 months of age. Speech samples were collected from 10 children with normal hearing and 10 children diagnosed with hearing loss before 6 months of age, and all children with hearing loss received amplification and intervention services. With the exception of one child with normal hearing, all of the participants had expressive vocabularies of fewer than 10 recognizable words, as measured by the Language Development Survey, a standardized parent report of vocabulary. According to a procedure for determining socioeconomic status, it is determined that the participants generally came from homes where the primary income earner had a college education. The procedures are thoroughly described in the published article of the study. Each child was recorded while playing with a standard set of toys sitting on the floor with a parent. Most of the recording sessions lasted 20 minutes and captured high quality video and audio. Speech production was assessed in 2 ways. First, broad segmental transcription using both perceptual and spectrographic information, and second, quantitative analyses of the speech spectra themselves. Bias was reduced by using a broad segmental transcription aided with spectrographic displays, and the transcriptions were used to assess syllable shape and consonant type. The transcription of consonants was in terms of manner, such as stop, fricative, affricate or trill, and place. Utterance shape was derived from the transcriptional data. Children's speech production was also measured and characterized by acoustic measures of formant frequencies. A detailed set of acoustic measures was made on a subset of the utterances identified in the transcriptional analysis, and both a spectrogram and a spectral cross-section were used to estimate F1, F2, and F3. The authors performed checks of reliability and determined the amount of difference between analysts to be acceptable. The results in terms of syllable shape, it was found that speech production of children with hearing loss contained fewer multisyllable utterances with consonants than their normal hearing peers. The study's results for syllable shape compare well with previous research, which states that canonical babbling, which involves the production of CVs, was delayed for children with hearing loss. In terms of consonant types, it was found that speech production of children with hearing loss contained fewer fricatives and fewer stops with alveolar stop placement. It is noted that children with normal hearing were more likely to produce stops with alveolar stop places of articulation and were more likely to produce fricatives than children with hearing loss. In terms of vowel formant frequencies, the acoustic analyses revealed that both the normal hearing and hearing loss groups have a normal variation in F1 for their 12 months of age, but the hearing loss group did not move as far from the mean F2 as often as the normal hearing group did, as measured by average standard deviations. So in articulatory terms, the front-back tongue movement for children with normal hearing was more likely to be more distant from mid-position than for children with hearing loss, particularly for high vowels. There appears to be no differences in the use of tongue height between these groups of children. In conclusion, these analyses are evidence that the speech production of children with hearing loss is different from that of children with normal hearing at 12 months of age. The differences are apparent in the number of multisyllable utterances with obstruent or nasal consonants. The differences are also apparent in fricative production and place of articulation for stop consonants. The results of this study suggest that researchers need to ensure that early intervention provides sufficient opportunities to hear the speech of others. Intervention should emphasize on providing examples of complete language samples so that children with hearing loss have access to a range of production. Future studies on this topic may be explored via a longitudinal study of these participants as they acquire speech. Speech samples will need to be analyzed with the same acoustic parameters and more refined transcription as they grow older. Acoustic analyses in this study and in previous studies are providing meaningful information in understanding the differences in children with hearing loss and children with normal hearing more thoroughly than transcriptional analysis alone, even at 12 months of age. In terms of the purpose of this study, it was clearly and concisely summarized. There was a logical and convincing rationale, there was a current, thorough, and accurate literature review, and the purpose, questions, and hypotheses were logical extensions of the rationale. The sample size and selection criteria of participants were adequately and clearly defined. Instrumentation for data collection were appropriate, and the researchers presented evidence on reliability and validity of data collection methods. The procedures were adequately outlined and appropriate for the research design. Also, the data analysis and statistical methods were clearly described. The results of the study were clearly related to the research problem, and the organization of data was clear and appropriate. The conclusions were drawn directly and fairly from the results, and suggestions for future research were identified. Overall, this was a strong research design. It can be applied to clinical practice by prompting clinicians to reevaluate their approaches to early intervention for children with hearing loss in order to better facilitate speech development.